Good morning. I hope all are joining still.
Morning, ma'am.
Morning. May I know your name? Who are you? Tell me your name.
Yeah, ma'am. Myself, Prathamesh.
Your name?
I'm Prathamesh.
Prathamesh. Where are your friends?
I'm there coming soon.
They will come, sure.
Yes, ma'am.
Okay. Are you regularly attending, Prathamesh?
Yes, ma'am.
What are the problems you are facing? Is connectivity all right throughout the class? Are you able to hear?
Yes, ma'am. Clearly.
So you will be continuously there. What was my last class? Can you tell me? Prathamesh, left. What was my last class, Prathamesh? So, okay, I hope all of you are there. You will try to understand what we are talking. And anyway, there is no alternative. We have to continue with this aid of teaching. So, today I am going to discuss about the remaining aspects of the competency, that is BI 6.5, which is related to the role of vitamins in metabolism and homeostasis. So we were trying to understand, we have finished about all the aspects related to fat soluble vitamins. In that, details of vitamin A, D, E and K we have learned. Now we are going to understand, already I had started with some of the water soluble vitamins also in alignment with the hematopoietic system and all. We have learned the role of vitamin B9 and B12. Also about vitamin B1 in the series of water soluble vitamins we have dealt in detail. So today I am going to deal in details about. So I have completed under this water soluble vitamins this vitamin B1, that is thiamine, riboflavin and niacin I am going to deal today. Next class I will be dealing about these two vitamins, then remaining aspects of the other vitamins and details about ascorbic acid will be discussed in one class. So now remaining 4-5 classes I will be dealing with the remaining vitamins under vitamin B complexes and the ascorbic acid in detail. So today we are going to start with the riboflavin, that is vitamin B2. So this vitamin was first time isolated by the scientist Alexander Blake in 1872 from milk, and as it is having a slight yellowish in color and it was derived from the milk source, the vitamin was named as lactochrome or lactoflavin. This was the name given by this given to this vitamin in the beginning. So next, later on it was called as riboflavin. So this is a yellow compound, that is why flavus means yellow in Latin. So the name was given as riboflavin. It can be obtained from different sources like egg, that will be called as ovoflavin, milk source if it is there, lactoflavin, like that. Depending on the source, it can be named further. So it is a simple vitamin that we are going to understand. and it is energy liberating vitamin. So it is relatively heat stable but decomposes in the presence of visible light. That means it is damaged by the exposure to light. So the structural details of this vitamin, it has got isoalexazine ring with a ribitol, that is a sugar alcohol, as a side chain. So this is basic ring of isoalexazine, and to which this sugar alcohol, that is ribitol, is attached, and that gives rise to this whole structure of riboflavin. So you have to remember two components, isoalexazine ring with ribitol. As we know, we try to understand the sources of these vitamins. So these are the sources of vitamin B2, which are liver, dried yeast, egg, and whole wheat. These are the rich sources of vitamin B2, whereas fish, whole cereals, legumes are and green vegetables give good sources of this vitamin. So here you can remember the sources of vitamin B2. Now this vitamin is mainly required with the metabolism of carbohydrates, and hence the requirement of this vitamin will be in relation to the calorie intake in the form of carbohydrates. So it is directly related to the calorie intake. So on an average, adults on sedentary work require about 1.5 milligram per day of riboflavin. And as you know, during these physiological stages of pregnancy, lactation, and with aging, there will be additional requirement, and that is up to 0.2 to 2.4 milligram per day additionally to the daily requirement is to be added. Now coming to an important aspects of its biological functions. So there are two coenzyme forms of riboflavin. It exists in its active forms. They are called as flavin mononucleotide and flavin adenine dinucleotide. So they are necessary for oxidoreductase enzymes. So they are the prosthetic groups, FMN and FAD. They are prosthetic groups of oxidoreductase enzymes. These enzymes are called as flavoproteins, which contain flavin mononucleotide and flavin adenine dinucleotide as the prosthetic group. These type of enzymes are called as flavoproteins, and you are going to learn these things in the biological oxidation. Next coming class of biological oxidation, you will have all these things together. So how it is formed from riboflavin by the action of flavokinase in presence of ATP, you get flavin mononucleotide. This flavin mononucleotide by the enzyme FAD synthase, that is again in the presence of ATP, will be converted into flavin adenine dinucleotide. So this is a mononucleotide, this is a dinucleotide. So like this, with the help of these two enzymes, flavokinase and FAD synthase, you get these two coenzyme forms of riboflavin, FMN and FAD in presence of ATP. So these are the structural details of these two, which you are not supposed to. So these are involved mainly in the oxidation reduction reactions. It is because these are the places where the hydrogen atom can be taken up by the FAD. So here in this ring you can see this nitrogen, these two are involved in the acceptance of the hydrogen molecule, and the FAD is converted, D is converted into its reduced form, that is FADH2. This is what is going to happen during the transport of electrons in the electron transport chain and other oxidation reduction reactions. You will come to know about this reduction reactions. So this is by FAD. So these two of the alloxygen ring, these two nitrogens N1 and N5 are able to accept the hydrogen atom. So with single hydrogen it is a semiquinone form, with double hydrogen it is a hydroquinone form. So these are the ways how these are going to act as a prosthetic group for oxidoreductases. So they play role in acceptance of the hydrogen atoms. So which are the reactions that are dependent on FMN and FAD as the coenzymes. So the enzymes which are involved in the oxidation of amino acids. So there are two reactions you have to remember which are FMN dependent. So during the amino acid oxidation, FMN is reduced to FMNH2 and it is reoxidized to molecular oxygen to produce hydrogen peroxide. So this you will learn in biological oxidation. Also in the respiratory chain, the complex 1, which is nothing but the NADH dehydrogenase containing NADH CoQ reductase enzyme contains FMN. So these two reactions are dependent on FMN as a coenzyme. This you can see in the electron transport chain, NADH to FMNH2 and to coenzyme Q. So the flow of electrons goes like this, and that is mainly involving these dehydrogenase enzymes. Apart from that, the reactions that are involving FAD, FMN we have seen amino acid oxidases, which will convert amino acid to keto acid and free ammonia. During amino acid metabolism, you will come across this reaction. FID as a coenzyme in various metabolism, these are the several examples of metabolic reactions dependent on FAD. So you have to remember these important reactions as an example for FAD dependent reactions in the metabolism of different biomolecules. So in the carbohydrate metabolism, the reaction catalyzed by the enzyme pyruvate dehydrogenase complex, PDH complex, you might have come across in the carbohydrate metabolism if it is started. So conversion of pyruvate to acetyl CoA by PDH is dependent on this FAD. So this is a component of pyruvate dehydrogenase complex. Next, in the TCA cycle or Krebs cycle, conversion of alpha ketoglutarate to succinyl CoA and succinate to fumarate. These two dehydrogenases require FAD. Acyl CoA to unsaturated acyl CoA in the lipid metabolism. In the amino acid metabolism, again, D-amino acid to alpha keto acid, D-amino acid oxidase. This is L-amino oxidase, which is FMN dependent, but FAD dependent are D-amino acid oxidases, which converts amino acid to alpha keto acid and ammonia is liberated free. Glycine to by glycine oxidase converted to glyoxylate and releasing ammonia. So this is the you will learn under glycine metabolism. Next is during the even in the purine metabolism, conversion of xanthine to hypoxanthine by the enzyme xanthine oxidase. This is dependent on FAD. Another example we have come across while understanding vitamin A, that is conversion of retinol to retinoic acid by cytosolic retinol dehydrogenase. This is also an example where you will find the reaction is dependent, this dehydrogenase also is dependent on FAD. So these are the biochemical reactions which require FMN and FAD as cofactors. So you have to remember these coenzymes role. This is the major biochemical role of these simple riboflavin, vitamin or vitamin B2. So you have to code these reactions biochemical functions of riboflavin. So deficiency, next is deficiency of this riboflavin. As such, in humans, it is uncommon because riboflavin is in some part synthesized by intestinal bacteria also. Its deficiency usually occurs in association with deficiency of other B complex vitamins, and the manifestations of this will be affecting mainly the tongue and lips. So characteristics like glossitis, angular stomatitis, cheilosis, pharyngitis and genital dermatitis are the major um, characteristics of riboflavin deficiency. So glutathione reductase activity, which is dependent on FAD, can be taken as a assessment to test for riboflavin deficiency. So you can see the images of the patient suffering from glossitis, magenta colored tongue and cheilosis in the riboflavin deficiency. Then this is the circumcorneal vascularization seen in the deficiency of this riboflavin. Irregular ridges in the nails in riboflavin deficiency you can see here. So these are the deficiency manifestations of riboflavin, which can be summarized as glossitis, that is related to tongue, magenta colored tongue, cheilosis, lip or fissured lip, angular stomatitis, that is inflammation at the corners of mouth, circumcorneal vascularization, proliferation of bulbar conjunctival capillaries is the earliest sign of this deficiency. Next, normochromic normocytic anemia and irregular ridges in the nail. These are the deficiency manifestations of riboflavin. To treat the deficiency, it is therapeutically can be administered in the doses of 5 to 10 milligram per day. The deficiency assessments, as I have already quoted, one of the enzyme, that is erythrocyte glutathione reductase activity with and without adding FAD can be checked in the laboratory. Apart from that, fluorometric assay of riboflavin in the blood also is a test for assessing the riboflavin deficiency. So where these methods you will come to know about the levels of riboflavin in the patient's blood sample. Then some of the examples which are structural analogs of this vitamin can act as antivitamins. The examples can be given as galactoflavin, for flavin monosulfate, a competitive inhibitor of FMN. Isoriboflavin and diethyl derivatives of riboflavin are also found to be the antagonists of with these vitamin riboflavin. So all these can be the structural analogs of them, and that will affect the vitamin activity, and thus they act as a antivitamins or antagonists for this riboflavin. So this is all about the riboflavin. You can remember simply to revise, you have to recall about its structure, sources. So basically it has got this ring with ribitol, that is sugar alcohol, isoleucine ring with ribitol. The sources, richest being yeast, egg, liver, milk are the rich sources. Requirement 1.5 mg and dependent on the calorie intake. Riboflavin has got two forms, FMN and FAD. These are mainly involved in the oxidoreductase enzyme coenzymes. Next, they are the examples of FMN dependent and FAD dependent reactions in the metabolism of all the biomolecules, carbohydrate, TCA, lipid. These are the specific steps where you will come across the role of these deficiency manifestation as glossitis, cheilosis, stomatitis, circumcorneal vascularization can be given administered orally in the doses of 5 to 10 milligram per day. Deficiency assessment is by fluorometric assay of the riboflavin and erythrocyte glutathione reductase activity. Antivitamins, that is some structural analogs, can inhibit its activity. They are the example. Coming to the next in the list, that is vitamin B3, also called as niacin, or this niacin is a general name for the nicotinic acid and nicotinamide, which may act as a source of this vitamin in the diet. So, it is obtained through diet in the two forms, nicotinic acid and nicotinamide. So this niacin, nicotinic acid, niacinamide and pellagra preventive factor of Goldberg are the names of vitamin B3. Remember, all these are the same names, or they can be called as a synonyms. Niacin, nicotinic acid, niacinamide and pellagra preventing factor of Goldberg. Why this name is given? It is because of its deficiency manifestation of vitamin B3 is pellagra. To treat this pellagra, this B3 is given, and that's why it is called as pellagra preventing factor of Goldberg. This vitamin is plays an important role in energy releasing metabolic pathways as well as in DNA repair. So it has got these two important roles. So it is coenzyme for the energy releasing metabolic pathways. Now in the beginning of classification of water soluble vitamins, we have seen they can be categorized into two different ways. One is hematopoietic vitamins and the other are metabolic, this either energy releasing vitamins. So hematopoietic being vitamin B9 and B12, all remaining water soluble vitamins of B complex come under the group of energy releasing vitamins. So this is also an important example or important vitamin for energy releasing metabolic pathways. So this niacin and they are all synonyms. This niacinamide is the active form of vitamin present in the tissue. The term nicotinic acid is should not be confused with nicotine. So this nicotine, which is obtained from the tobacco, which is having potent dangerous or toxic effects, whereas nicotinic acid is a vitamin which is of niacin or vitamin B3. So remember these two important points. Nicotinic acid is a vitamin and nicotine is the potent poison in the tobacco. The structural details. So this niacin is a pyridine derivative, that is pyridine derivative, that is pyridine 3 carboxylic acid. This is at the third position carbon atom. It has got the carboxylic group attached. So this is the basic pyridine ring to which there is carboxyl group attached, and this becomes niacin, that is pyridine 3 carboxylic acid. This is the structure of niacinamide, which is obtained. This is another. This is niacin is present in this form and can be obtained through diet. In this niacinamide, when this amide is given, this amide form is called as niacinamide. This is nicotine, which is present in tobacco. This amide of nicotine is nicotinamide. So nicotinic acid is produced from the oxidation of nicotine. So these are the structural details of this niacin. The natural source of this niacin is dried yeast, rice, polishing, liver, peanut, whole cereals, legumes, meat and fish. And half of the requirement of this vitamin is met by the conversion of tryptophan to niacin. About 60 milligrams of tryptophan yields around 1 milligram of niacin. So this is the vitamin which is being synthesized from amino acid tryptophan. That you have to remember. So half of the requirement is met by conversion of tryptophan to niacin. So this you have to remember. So it is synthesized in the body from amino acid tryptophan. During understanding of tryptophan metabolism, you will come to know catabolism of tryptophan, that is by the kynurenine pathway. Tryptophan is converted into niacin. That we are also going to learn further. Normal this recommended daily allowance is around 20 milligram per day, and additional 5 milligram is required during these physiological stages. Now here we will try to understand how this is synthesized from tryptophan. So dietary nicotinamide, niacin and tryptophan contribute to the synthesis of NAD and NADP. So 1 milligram of niacin is formed from 60 milligrams of tryptophan. PRPP, that is phosphoribosyl pyrophosphate, and ATP provides ribose phosphate and AMP moiety for the synthesis of NAD, and glutamate donates the amide group. So NAD, this niacin and niacinamide, they are synthesized from tryptophan and glutamine. So this is the synthetic pathway, that is synthesis of niacin from tryptophan by the pathway kynurenine. So you have to remember this kynurenine pathway, how which it goes from tryptophan to formyl kynurenine, kynurenine to 3-hydroxykynurenine, hydroxyanthranilic acid. This is semialdehyde and quinolic acid. This quinolinic acid is converted into NAD. So this is the pathway in the catabolism of tryptophan or the substances synthesized from tryptophan, you can remember. So this by the different enzymes, mainly kynureninase, it will be converted into quinolinic acid and NAD. So this is the metabolic synthesis of niacin from tryptophan. Now coming to its biological functions, the coenzyme forms of this niacin, they are called as nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. So this phosphorylation occurs at this hydroxyl group. So this NADP will be formed by addition of phosphoric acid residue to the hydroxyl group shown here into the ring of adenine mononucleotide. So this is nicotinamide mononucleotide. These two club together to form adenine dinucleotide. Di means two nucleotides are linked together, and this dinucleotide, when there is addition of a phosphoric group to this moiety hydroxyl group here, it will be converted into NADP. So this whole structure is of NAD. After addition of phosphoric group to this moiety, it will be converted into NADP. So these are the two important coenzyme forms, NAD and NADP. Many reactions depend on this NAD and NADP, and they are mainly oxidation reduction reactions. There is fundamental distinction between NADH and NADPH. This NADH is generated in various catabolic pathways, is oxidized in respiratory chain to generate ATP. NADH plays vital role in cellular respiration, whereas NADPH serves as a readily available reducing power in the biological system, and this is used as a reductant in biosynthetic processes. Remember the roles of these two important reducing equivalents, NADH and NADPH. So NADH is during catabolic pathways, and NADPH is utilized as reductant in biosynthetic processes. And this is the important. This is NADH is mainly in the cellular respiration, NADPH in biosynthetic processes as reductant. Now this will accept, this is ribose phosphate, and this NAD, there will be reduction by acceptance of two hydrogens and one hydrogen at this point. So NAD will be converted into NADH and H+. So the mechanism of oxidation reduction, as we have seen, XH2 gives NAD2, XH2 accepts NAD from this hydrogen from this substrate and will be converted into NADH and H plus is released free. So this NAD oxidized form, when accepts the hydrogen, will be reduced to NADH plus H plus. So always it is written like this. Reduced form of NADH is written like this always. Now, the enzymes which are dependent on NAD as coenzymes, they are involved in all the biosynthetic processes of carbohydrates, lipid metabolism. So, in the carbohydrate metabolism, you have the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase, which will convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. That is the reaction that you have learned under glycolytic pathway. So this reaction is dependent on NAD. So on the arrow you can show here the conversion of NAD to NADH. So lactate will be converted to pyruvate, that is in the anaerobic glycolysis. Pyruvate to acetyl-CoA by PDH complex. This is a link between glycolysis and Krebs cycle. So this table itself is going to give you the details about the reaction and where it is occurring, in where this step is occurring in which metabolic pathway, which is dependent on NAD. This is in carbohydrate. Now these are the reactions in lipid metabolism. During the beta oxidation, hydroxyacyl CoA is converted into beta ketoacyl CoA. Hydroxybutyrate converted to acetoacetate during the ketolysis. Next, amino acid metabolism, glycine to carbon dioxide and ammonium by the glycine cleavage system. Alpha keto acid of branched chain to respective acyl-CoA, degradation of valine. Krebs cycle isocitrate to oxaloacetate, alpha ketoglutarate to succinyl-CoA, malate to oxaloacetate during the TCA cycle. And in another example can be quoted as by the enzyme alcohol dehydrogenase during the metabolism of ethanol to acetaldehyde. In the alcohol metabolism, it is also dependent on NAD. So all these are catabolic pathways. Now NADPH generating reactions. So this glucose 6-phosphate to 6-phosphoglucanate, that is during HMP shunt, whether it is over I don't know in your carbohydrate metabolism, this HMP shunt is over. So main purpose of HMP shunt is generation of the reducing equivalent in the form of NADPH. So this reaction, glucose 6-phosphate to 6-phosphogluconolactone will be generating this NADPH. So 6-phosphogluconate to ribulose 5-phosphate, malate, and these are the two HMP shunt reaction. Another example during transport of acetyl-CoA from mitochondria to cytosol, that is malate to pyruvate by the malic enzyme, it will be converted formed isocitrate to alpha ketoglutarate during cytosolic isocitrate dehydrogenase. So these are the reactions involving generation of NADPH. So this is NADP, you have to show converted to NADPH. Now these were the reactions where you have seen production of NADPH. Now in the reactions where NADPH is utilized, the examples are given here. So they are all biosynthetic pathways of lipid, bile acid, phenylalanine, vitamin, folate, hemoglobin, and synthesis of calcitriol, that is vitamin D. So these are the reactions, specific reactions of these pathways which are dependent on NADPH. So this beta ketoacyl enzyme converting to beta hydroxyacyl enzyme, this ketoacyl reductase, enol reductase, all these reductases, hydroxylases are dependent on NADPH as the coenzyme. So in the biological functions of this niacin, you can see its role in all the different types of the metabolism. Carbohydrate, lipid, amino acid, again miscellaneous, that is alcohol. Generation is dependent on HMP shunt, during transport of acetyl-CoA to mitochondria, conversion of isocitrate to alpha-ketoglutarate. So it is necessary for the metabolism of all the biomolecules, either in the form of NADP or NADPH. It is used for synthetic as well as catabolic reactions. So deficiency manifestations will involve all the mm, all the tissues and organs which are dependent on these formation of these biomolecules. So the deficiency manifestation of this next aspect is the causes for niacin deficiency. Now first and foremost, as we have seen that it can be synthesized from tryptophan. So dietary deficiency of tryptophan will lead to deficiency of niacin, because we have seen about half the requirement is sufficed by the tryptophan synthesis from tryptophan, that is around 60 milligram of tryptophan will yield it yield milligram of niacin. Do you remember? So 60 milligram of tryptophan yields around 1 milligram of niacin. So of tryptophan is going to cause deficiency of niacin also. What other causes may be there? So, this tryptophan deficiency can be seen. This pellagra is, that is the deficiency manifestation of niacin. It is mainly seen in the people whose staple diet is maize or sorghum, that is jawar or guinea corn. So this sorghum contains leucine in high quantities. Leucine inhibits the phenylalanine phosphoribosyl transferase, that is QPRT enzyme in the synthetic pathway from tryptophan to niacin. So niacin cannot be converted to NAD leucine pellagra, and that is why it is called as due to the deficiency of tryptophan. So there will be manifestation of deficiency of niacin, that is pellagra. Next is deficiency of the enzyme which is involved in the synthetic pathway, that is kynureninase, that is the important enzyme in the pathway of tryptophan to niacin, and this is pyridoxal phosphate dependent, that is vitamin B6 dependent. So the conversion of tryptophan to niacin is not possible in pyridoxal deficiency. So deficiency of one vitamin leads to deficiency of other. That is mainly explained by the deficiency of vitamin B6. We are going to learn tomorrow. So this vitamin B6, the active form of which is pyridoxal phosphate. So if B6 is not there, its coenzyme form will not be there. So this enzyme will not be acting. So the important enzyme kynureninase in the deficiency of vitamin B6 will not go or cannot synthesize niacin by the tryptophan, and hence the deficiency will be manifested, that is indirectly deficiency of one vitamin manifests the deficiency of other vitamin. So vitamin B6 deficiency leads to vitamin B3 deficiency. Got it? So conversion of tryptophan to niacin is not there in the pyridoxal phosphate deficiency. This pyridoxal phosphate deficiency, hence in the deficiency of vitamin B6, niacin deficiency also will be there. And you can now imagine that deficiency manifestation of vitamin B6 will have the features of niacin deficiency also. So this is interdependence of one vitamin in other. Next is the treatment by the drugs like isoniazid. Mainly this is a anti-tubercular drug. So for the treatment of tuberculosis, this again INH is given, and this INH is going to inhibit pyridoxal phosphate again. So during the treatment of tuberculosis by the drug INH, there will be inhibition of pyridoxal phosphate formation, and that in turn by the same mechanism is going to have deficiency of niacin. Are you getting? So during the treatment of tuberculosis by isoniazid, this is the inhibitor of pyridoxal phosphate. So, as pyridoxal phosphate is not there, niacin also will not be synthesized. So, this drug is going to cause niacin pyridoxal deficiency. This deficiency in turn is going to cause niacin deficiency. So these are the causes for deficiency of niacin. Apart from that, the tryptophan absorption from intestine by these transporters is defective in the disease called as Hartnup disease, and hence there will not be the synthesis of niacin. So another disorder or disease called as Hartnup disease. You are going to learn this in detail in amino acid metabolism. So tryptophan absorption from intestine is defective in the people who are suffering from Hartnup disease because of the defect in that transport protein itself, and hence there will be deficiency of tryptophan, and that deficiency is going to lead to niacin deficiency. Also, it is seen in this Hartnup disease, tryptophan is getting excreted in urine in large quantities. So, this is another cause for niacin deficiency. Next comes the another cause, that is carcinoid syndrome. So, this is a type of tumor. So, the formation of the tumors utilizes major portion of available tryptophan for synthesis of serotonin, and hence tryptophan is not available for synthesis of niacin. All these points, these two important points, you will come or understand much better when you understand the tryptophan metabolism. So carcinoid syndrome and Hartnup disease, you will be coming across again in the tryptophan metabolism. Here we are taking into account as a cause for niacin deficiency because tryptophan is a precursor for synthesis of niacin, or niacin can be synthesized from tryptophan, and hence wherever there will be tryptophan absorption defect or tryptophan is utilized for synthesis of some other compounds, that time there will be deficiency of, parallelly you can see deficiency of niacin because synthesis is decreased in those conditions. This is what is about the causes for niacin deficiency or vitamin B3 deficiency. So they can be because of dietary deficiency, that is those who are continuously eating maize or sorghum that contains leucine. Leucine inhibits this QPRT enzyme, so niacin will not be synthesized. Next is deficiency of one vitamin, that is B6, will inhibit this kynureninase. Again, the treatment with this drug inhibits pyridoxal phosphate. This pyridoxal phosphate inhibits kynureninase, or deficiency of this pyridoxal phosphate will hamper the functioning of kynureninase, no synthesis of niacin. And these are the two disorders related to tryptophan metabolism or the production of other compounds from tryptophan. They are going to affect the synthesis of niacin from tryptophan. Thus, these are the causes for niacin deficiency, and the deficiency manifestations of niacin are manifested as pellagra. So this is a classic disease caused by deficiency of niacin, which mainly affects the patient's skin, GI tract and central nervous system. The disease is mainly characterized by 3Ds. You have to remember this classic uh, characteristic feature of pellagra, that is 3Ds. manifestation, that is like dermatitis, diarrhea and dementia. And if untreated, it progresses to the fourth D, that is death is inevitable. So you have to identify and treat it properly to prevent the fourth D progression. That is, symptoms of this pellagra are. Now let us understand one by one why dermatitis, diarrhea and dementia is seen in these patients. Dermatitis is mainly in the feet, ankles and face. Increased pigmentation around the neck is known as the castle's necklace is seen in the patients suffering from pellagra. Nails have got irregular surfaces. So these are the deficiency manifestations seen in the patients of the pellagra. So this is a severe, you can see chronic deficiency will look like this: pigmentation around the neck, the areas where there is exposure to sun. So they will be like sunburns. So they are this like sunburns. There is a bright red erythema and pigmented skin rash over areas exposed to sunlight, like neck, ankles and back of the feet. So neck, ankles and back of the feet, there will be bronzing and thickening of the skin that gives appearance, and classically it is called as castle's necklace. So these are the pictures of niacin deficiency. Niacin deficiency pictures, skin lesions seen, and this deficiency manifestation in nails as irregular or uneven surfaces. So some patients will have such uneven surfaces of the nails. The skin lesions may worsen by formation of vesicles and bullae. So if it is not treated, the patient may lead to the death. Next symptoms, that is diarrhea. This is mainly deficiency of niacin causes inflammation of the gastric GI tract, leading to loose stools and diarrhea with blood and mucus, which may lead to the rigorous weight loss. And there is also associated nausea, vomiting and glossitis seen in these patients. The third characteristic feature is the dementia. Dementia is mainly seen in the patients who are suffering from chronic deficiency of niacin. So in chronic cases, dementia is seen, and in acute condition of pellagra, it is delirium. And the mild symptoms will be irritability, inability to concentrate and poor memory are more common in mild cases. Ataxia and spasticity also is seen. In the severe cases, there is posterolateral tract degeneration, ataxia, spasticity, involvement of bladder and rectal sphincter, depression and depressive psychosis, that is leading to the features of the one of the D, that is of pellagra, that is dementia. So these are the three important niacin deficiency features. It is a pellagra which is characterized by dermatitis, diarrhea and dementia. The most common thing during your vivas will be asked about the deficiency manifestation of niacin, the general term, that is the pellagra and its characteristic features. During the quiz or in MCQs, you may be shown these pictures of deficiency manifestations of all the vitamins what I am showing you during my uh, detailed talks. So you have to remember these pictures. It can be asked in the form of MCQ also. So that way you have to know about the deficiency manifestation of these different vitamins. Also in Google you can get variety of the pictures, but all the textbooks, whatever pictures we are having in the textbooks, that only I am going to give you. Next, how to assess the deficiency of niacin. It can be done by assaying the urinary excretion of N-methyl nicotinamide, that is derivative of this nicotinamide, and that can be done by spectrophotometric method using CNBR as a reactant. Another is by microbiological method That is done by assessing the growth of Lactobacillus arabidosus in a medium containing niacin. So these are the two methods for assessment of niacin deficiency. Some of the structural analogues act as an antagonist to niacin. They are pyridine, 3-sulfonic acid, 3-acyl acetylpyridine. They are the structural analogues and they can act as antagonists to niacin. Niacin can be given in the doses of 15 to 20 milligram thrice a day orally. So this is used for its treatment of the deficiency. Whereas therapeutic uses of this niacin, so the format it has got an important role in the pharmacological dosage of 2 to 4 gram per day. This niacin helps in inhibition of lipolysis in the adipose tissue and decreases the circulating free fatty acids. It reduces triacylglycerol synthesis in liver. Nicotinic acid reduces serum cholesterol level and hence it is used in the treatment of hyperlipoproteinemia. In high doses, niacin is useful to reduce the lipoprotein A levels in the blood. So, in the patients who are having hypercholesterolemia and had heart attack or related cardiac disease, in them this niacin is given as a in the pharmacological dosages that will help in reducing the levels of LPA, that is lipoprotein A, which is found to be culprit in cardiovascular diseases. So this nicotinic acid, if it is given orally or parenterally, produces a transient vasodilation of the cutaneous vessels and histamine release, and hence the reaction is accompanied by itching, burning and tingling. So a overdosage also has to be monitored. So under the strict physician's guidance only you have to take this niacin. So this is all about the second vitamin in the today's list that we have seen. Important reactions of niacin, coenzyme D, reducing, and then reduction reactions, mainly dehydrogenases, reductases. Deficiency causes, deficiency manifestation of this niacin, all the 3Ds, and if not treated, the fourth D is there assay, antivitamins and therapeutic uses of niacin. So all these aspects you have to remember about this vitamin B3, that is niacin. So you can make out your own mind map or a chart to remember the important points related to the vitamins. Okay, so with this I come to the end of today's class. At the end of the classes, I will give you the cases related to vitamin deficiencies, and also MCQs will be discussed, or I can send you vitamin related MCQs. So till you are here, follow these guidelines for prevention of the COVID-19. Stay home, stay safe. Okay. Thank you.